Good afternoon, everyone. I'm Kira Curtin, Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is a multidisciplinary study links androgen signaling with severe COVID-19 symptoms in men, and is sponsored by Lexogen. Our speakers today are Hani Goodarzi, an assistant professor at the University of California, San Francisco, and Martina Sauert, a product manager at Lexogen. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Hani.
Thank you so much. Good morning, everyone. Let me start by a quick introduction. I'm Hani. I'm an assistant professor at UCSF. I've been here for about 6 years now. And let me preface by saying that this is a multi-lab, multidisciplinary research that I'm going to present today. And was a very deep collaboration with a few other folks at UCSF and elsewhere that I have been working with on this project. So I was a contributor, and based on that, I think I wouldn't know necessarily all the answers to all your questions. It's a very sprawling research question, so I just wanted to start with that disclaimer. And as I mentioned, this was work that really started by my colleague Farnak Fatahi here at UCSF, really early on during the lockdown, the first lockdown that we experienced at UCSF right after the pandemic. She really rolled up her sleeves and rallied us to actually take on this important question. So the first question that we tried to ask is basically, what are the molecular determinants of ACE2 expression? As we know, this is the receptor of SARS-CoV-2, and it actually shows very interesting and intriguing tissue-specific patterns of expression in both men and women. And you can actually look at basically the expression patterns across different tissues and specific cell types. So this is far from a universally expressed gene, and it actually shows very intriguing patternings across different cell types. But as you can see, these there's specific cell types that show very strong expression of ACE2, and we chose to focus on heart. Rather, Farnak chose to focus on heart as a model for ACE2 expression because it is fairly highly expressed in this model, so it's easier to work with, but also a very well-established protocol exists for differentiating human embryonic stem cells into cardiomyocytes. So again, Farnak is the stem cell researcher. She knows how to do these things. I don't, but basically there is a set protocol that with very high fidelity, and you can derive faithfully cardiomyocytes from human embryonic stem cells over a period of time. This is a result from that effort. As you can see, very solid ACE2 expression in these models. Now, what you can do with having access to these really precious, specific cell type models, is that you can actually do drug screens. So there are multiple uses of drug screens that you can think of. One use would be the actual effort of drug development, trying to find new drugs that do what you're looking for. But you can also use established drugs to understand biology. Basically look at the impact of compounds that we very well know and understand to really interrogate the biology of the system. So in this case, we are doing a combination of the two. But really, Farnak started with a screen, a systematic screen of about 2,000 FDA-approved compounds, basically treating the cells, cardiomyocytes, with these drugs, as I mentioned, about 2,000, and then measuring ACE2 expression after treatment. So I'm showing you here 3 examples. There's no treatment. This is the baseline expression of ACE2 in these models. This drug, you see higher expression. With this drug, you see lower expression. So basically, you will get both ends of the spectrum like any other screen. So this is the really a simplified output of the results. Once you've normalized across plates and across experiments, you basically associate what we call a normalized score with each of the tested compounds, and the colors basically denote the ones that are more or less significant, and the direction is basically whether they're down-regulating, the treatment is down-regulating ACE2 or up-regulating ACE2. And again, I'm showing you as bar graphs individual sets of up or down regulators, basically. So again, emphasizing that these are normalized z-scores that measure how much increase or decrease in ACE2 expression we see after this treatment. So this is basically where my lab and our expertise come into play. The question of where do we take this next? So first, we'll take these individual hits out and validate them, because you don't want to just rely on this screen on a high throughput format. So these are results of those validations in 2 different concentrations for these drugs. Again, more or less validating across the board all the hits from the original screen. So really emphasizing and highlighting that the original screen was really actually a high quality one, and we can go ahead and use that data. So one of the first things to realize is that a 2,000 compound library is not a lot of compounds. So one of the questions that we have been working on in our lab is basically the question of how can you translate more limited cell-based screenings that are done in vitro to much larger and scalable drug screens that can happen in silico, basically your computer. So one of the underpinnings well, first of all, I should mention that this question has been around for quite some time, and I, as a scientist, have been working on it for most of my career in research, for that matter. But the reality is that, for the most part, computational models had lagged behind in kind of performance in this kind of task. But very recently, advances in deep learning and more sophisticated computational modeling has actually allowed us to tackle this problem a lot more effectively, which is the direction that we are looking at, and we are quite excited about. So one of the key aspects of these kinds of deep learning models is the idea of an architecture of neural networks, which are called graph convolutional neural networks. I don't have a lot of time to go into the details of how they work here, but basically the basics is this. If you have a molecule, you can basically represent it as a graph, and what the model after training actually is trying to do is basically find features, more or less, in this graph that are informative of what we are trying to measure. So this is basically the very simplified architecture of what our model would look like. So on one end, you will get this graph representation of your compound, and in our case, we'll have 2,000 of these, more or less. And so the model is basically applying a graph convolution layer to this graph to find what we call an embedding of the molecule. So an embedding of the molecule is basically a multidimensional continuous space, basically. Each graph will go into a vector of values, as shown here. And these vector of values represent this molecule according to the model. And then the model is trained to in this case, perform a regression task, which is to predict the ACE2 expression. So basically, for every compound, you have as input the representation of the structure of that compound, and on the other end, you have ACE2 expression. And you're asking whether I can, across 2,000 compounds, actually build a model that, given the compound, can predict changes in ACE2. So the idea is that once you have a model that can accomplish this task, then you can actually use it on compounds that it has not seen before. So do in silico drug discovery, if you will. So first of all, the question is, can we actually do this? So we started, as I mentioned, with 2,000 compounds. This is not a lot when it comes to deep learning. This is actually a small number. We did a training validation test set split as well, which even makes them fewer. So in our initial attempts actually failed, again, just because of the sample size. But what really saved our bacon, if you will, was the fact there were actually a couple of tricks, but the key one was the fact that Farnek's group had, in addition to this ACE2 screen, really had generated quite a few other data sets using the same set of compounds. So this allows us to train a model that simultaneously analyzes all of those screens, not just ACE2, so be able to generalize and get a better understanding of association between molecular features and bioactivity, if you will. And we also took advantage of a number of other tricks as well to deal with smaller sample sizes, including regularizations and ensemble approaches. So in the end, this was the performance of our final model. As you see, the training goes fairly well. The validations and test sets are okay. So overall, it's doing pretty well. So the next question is, can we actually do this to look at not just the 2,000 that we were trained on, but over 10 million other compounds in silico? So the answer is simply yes. You can actually, once you have the model, you can basically run it on as many compounds as you want. Now, whether those results are valuable or not is a different question that we'll get to. But basically, the map on the left, the graph on the left, is a 2D representation of basically the compound fingerprints. In the UMAP space, a lot of you who have seen single-cell sequencing data, for example, or other transcriptomic or genomic data, you're familiar with this representation. It's just a visualization of the underlying data in the 2D space. And I've basically colored the FDA-approved drugs in orange here. The Zinc-15 library, which this is the main library in silico library that we are using. As you see, it has 10 million compounds that we used here. And then in red, we have colored our in silico hits. A couple of points here. FDA drugs are FDA-approved drugs are kind of, some of them are quite weird, and as you can see, there are these outliers here in the space. So, despite the fact that these might have been effective or not, the model will have a hard time generalizing to these. So most of the focus is here in the main blob. Again, this is very diverse. It's just in contrast to these outliers that it looks like a blob. In reality, this is a very diverse set. What is really important here is that the model is actually predicting different kinds of drugs. So in this space, the reds basically are not in one place; they're in different places. Again, we don't want to overemphasize this 2D representation, but the underlying data also shows the same thing. Now, what we can look at more closely is actually compare, basically in this space, pretty much compare and contrast the in silico drugs and hits we have found and the FDA-approved hits that we have found, and basically ask what is the relationship? What is the diversity of hits that we are getting from this in silico screen? So this is basically a blow up of this zoom in of this analysis. Every point is a compound, an in silico compound. Every rectangle is an FDA-approved drug, and all of these are hits, either for the FDA-approved compounds or in silico compounds. And they're colored based on the clusters. So we have done clustering on the underlying data. They're colored based on clusters. The idea being that the drugs that are in the same cluster tend to look similar, to have shared similar features, basically. Now, as you can see, again, there are a diverse set of drugs, but more or less we are seeing a fair bit of enrichment in a couple of categories that I'll actually come back to. Okay, so a couple of tests first. So as I mentioned, you have a model, you give it compounds, and it gives you a prediction. But whether that prediction is useful or not is something that we need to actually test outside of the test set that the model came with, basically. So basically another step in validation of the model. So to do this, we basically picked some of the basically hits that came out of the model here, significant hits, specifically 6 of them, and we tested them individually as well in comparison to the hits that came out of the screen. So as you can see, these are basically, we're measuring ACE2 expression in cardiomyocytes, where we are treating the cells either with the FDA-approved hits. These are the blue ones. You can see, as expected, they reduce ACE2 expression. Whereas, and if you look at the 6 compounds that we have found coded from A1 to A6, they're basically significantly reducing the levels of ACE2 as well. And more importantly, actually, quite a few of them are more potent than anything that was available in the FDA library. So this is really emphasizing that the model is actually doing its job quite well. So this was in cardiomyocytes. We even extended this to, rather, Farnack's group also extended this to primary epithelial cells, again, confirming that this happens in all models that have ACE2 expression, basically, both of these models that have ACE2 expression. So again, here in this case as well, if you treat the primary cells with these new drugs, you can reduce ACE2 expression. Okay, so in biology, when we train a model, train a computational model, part of the goal is to, of course, do a drug screen, for example, in this case, or make predictions. But a lot of the times, actually, there's another component, another side to model building, and that is trying to understand what the model is actually learning as a way of kind of like understanding biology. The idea being that a model that is learning biological input and output creates an abstraction of biological systems that are more simplified abstraction of biological systems. And by understanding that abstraction, we learn something about biology. So that's really part of what we do as computational biologists and model builders. So one of the questions that we would like to answer is that drugs have one-to-many relationships. So one compound can have drastic effects, can even bind multiple hits in the cell. So the first question is that we wanted to answer is that what are the underlying genes and subsequently pathways that are actually impacted by these hit compounds? Okay. So to actually answer this question, we built a statistical model that basically looks at a compound and likely genes and proteins that it hits. So this is basically makes it a gene level analysis. The idea being that if you have multiple drugs, let's say gene A is important, for what we are measuring, in this case, ACE2 expression. In that case, any drug that would inhibit gene A should give us a significant z-score. So by combining all the predictions of drug to gene mappings, drug to protein mappings, we should be able to do a gene level analysis that gives us a confidence interval around whether this gene is actually important or not, based on the profile of potential interactions with drugs. So that those interaction predictions come from established tools in the pharmacogenomic field. We used SEA, for example, which was developed by Mike Kaiser here at UCSF. So basically, you input to that model a list of compounds, and you get out of it a list of predictions for each compound. List of predictions of proteins that are going to be targeted by that compound. And then there's, again, one-to-many relationship, so you can tally those interactions, potential interactions, into a map that looks like this. And again, this is the part that we are interested in. These are the genes that are likeliest targets here based on our screen. And they're enumerated here individually as well. Okay. So basically what we can do in the end, we can generate that map. You basically use our models and use SEA to generate maps that look like this, that basically tells you these are the hit compounds, and for these hit compounds, what are the genes that were likely to be involved. And as you can see, there's some of these genes have very nice entanglement with the data. So you get this 2D map of not only understanding not only seeing which drugs were effective, but also understanding why they were effective. So of course, you can do this at the pathway level as well. So you can look at these, combine these gene level scores into pathway level scores. And for this, we take advantage of a tool that we developed it quite a while ago. It's called iPage, similar to other gene set enrichment analysis tools. But as you can see, these are the pathways that are significantly enriched among the target genes with lower z-scores in our screen. Now, what is really nice here is that, again, the FDA-approved library is only 2,000 compounds, is limited, but we can augment this with our 10 million in silico compounds. So we can do a similar analysis, exactly what I showed you for the FDA-approved library, but this time with the in silico hits, and try to look for the genes that they are predicted to be targeting and the pathways that they're predicted to be targeting. And as you can see, as expected, there is a fair bit of overlap between these two analyses. And two pathways really kind of like pop out that from this data, and I'm going to spend most of my time on one of them, and that one is androgen receptor signaling. So here we have basically highlighted in both of these pathways the genes in yellow, or rather the proteins and enzymes in yellow that were predicted to be hits by our drug screen. Okay, so where do we go from here? So we took a step back, and we now specifically focused on anti-androgenic drugs in our screen that were actually hits, and they were basically the basis of what we see here in terms of this map. So now that we have really established that this is really these two, but specifically androgen signaling is a specific pathway that may be involved based on our results and analysis in ACE2 expression control, we can basically take a step back and validate this hypothesis systematically. So first of all, making sure that this can be independently verified, and the responses shows dose dependence. Very important observations in both cases. So you have cardiac cells that are derived from two different cell types. And as you can see, in all of these conditions, more or less, we can confirm that treating the cells with anti-androgenic drugs, as expected, actually reduces ACE2 expression significantly. So those were the normalized values. You can also show this. I can also show you this with representative images. Again, ACE2 expression in green. You can see that the levels of green go down quite significantly. TMPRSS2 is also another component of this pathway, and again, you see that TMPRSS2 is also going down by and large. Okay, so this implies that there is a link between androgen signaling and ACE2 expression. So the first question we asked was whether we can actually corroborate this with existing data sets out there. So we looked at a data set that had done RNA-Seq in AR knockdown cells, and in comparison to, I've also integrated that with an AR ChIP-seq data that actually tells you where AR is binding across the genome. So here I'm first of all on the left showing you the volcano plot of the data we're starting with. So as I mentioned, it's an AR knockdown data set. So as you can see, very significant, reduction in AR expression as you would expect. Also TMPRSS2. On this side, you're seeing genes that are upregulated. On this side, genes that are downregulated. Actually, ACE2 is here. So ACE2 actually shows substantial and significant downregulation in this data set on its own. But you can actually broadly ask, do we see AR downregulation of the AR regulon in this data set? Again, as a confirmation, we see very, very nice enrichment of AR bound targets. And by target, we mean, promoter binding within 5 kb. There's a very nice downregulation of these targets, and then ACE2 is among those that are downregulated. So again, we corroborated this using a CRISPR-Cas9 model, where we are basically trying to knock down or knock out AR either in cardiac cells or primary cells. And in both cases, we see that reducing AR expression using these CRISPR-Cas systems also reduces ACE2 expression. Again, like linking that causality. Okay. So this is based on a link simply based on ACE2 expression on the surface of the cells or just broadly. Now the question is, will that actually have an impact on the SARS-CoV-2 interaction with the cell and infections, basically, which is really the guiding principle of this project. So one of the first steps we took to answer that was basically to repeat these experiments, but in addition to looking at ACE2 expression, we can also look at spike protein take-up by the cells as a measure of basically infection. And as you can see here, basically treating the cells with dutasteride, as I showed you earlier, reduces ACE2, and treating cells with DHD increases ACE2. So this is where we started. And concomitant to this, we can also basically see that the spike proteins are also showing the same pattern. So again, lower ACE2 expression reduces spike protein pickup, and DHD and increased ACE2 expression increases pickup. So basically showing that we are going down the right path, basically. Okay, so again, that was in cell models. We can confirm this, and the impact of these drugs on ACE2 expression in bronchial epithelial cells as well. Again, human primary cells from 3 donors. By and large, we can again look at the effect of these compounds and see that they do result in lower ACE2 expression. Okay, so this has created a molecular map for how we go from androgen signaling to ACE2 expression and infection. Of course, these infection models are very difficult to work with, and the cell-based models are a bit too simplified. So we actually took advantage of long organoid models to be able to create a more faithful kind of representation of what happens in the body actually during infections. So we again use embryonic stem cells to over a long period of time derive long organoids from embryonic stem cells. And here I'm showing you kind of like the makeup of these long organoids. As you can see, they have different cell types that you would expect to see, and again captures the diversity of a tissue basically. So first of all, are these human embryonic stem cell derived long organoid models of what we expect? The answer is yes. To do this, we took advantage of IF stainings and also taking many time points and doing RNA sequencing to specifically at this scale, we use 3' end sequencing to be able to look at generally at the different markers associated with different cell types and see when they come up again as over the time span of the differentiation. And as you can see, we are seeing the different markers associated with different cell types that we would expect to see as we get closer to the end of the differentiation protocol. And here, these are the genes that are, AR, ACE2. These are the genes that we're interested in. As you see, their expression is also present in these long organoid models. So these are basically organoid models that we can use to study infection. So first of all, having these organoid models allows us to actually take them into BSL facilities and they actually do the infections, and these are the results of those. First of all, again, in these long organoid models, if we use anti-androgenic drugs, we can reduce ACE2 expression. And concomitant to this, we can also reduce basically infection rates in long organoid models as well. Again, emphasizing that there is this link between ACE2 expression on the surface and the ability of SARS-CoV-2 to actually infect the cells. Okay. So again, one of the key steps in our approach to science always involves corroborating what we do in the lab in cell-based models or even mouse models with what happens in the clinic. So thanks to a couple of clinicians on our team as well, we could take a look at two places where data was available that we could try and corroborate this and see whether there is evidence for it in the clinical setting as well. So one was at the Yale Hospital, basically looking at if ACE2 is present in the heart and is basically hit by SARS-CoV-2, do we see an association with the same patients who have prostatic disease and who will have AR signaling dysregulation? And as you can see, we did in fact observe that association. And more importantly, we also looked at a much larger UK Biobank data that also showed basically androgen signaling associations with COVID-19 outcomes. And finally, more recently, there was actually anti-androgenic drug trial in Brazil, if I'm not mistaken, that actually corroborated this as well, that the fraction of patients who were admitted to hospitals that were COVID-19 positive was significantly lower in patients who were taking anti-androgenic drugs. Okay, so to summarize, I think this study really showcases how we approach science, really building models of biology and then interrogating them both using experimental measurements, but also sophisticated computational analyses, and then linking them back to basically biology of the disease and what happens in the clinic. We do this most of the time for cancer in my lab, and in Farinak's case, she studies more GI diseases, but the emergence of this pandemic really changed all of our lives, and this was our way of contributing to that effort and maybe gain some understanding of how the process works. And with that, I want to again thank everyone who did this work. I don't really think I was even the best person to necessarily present this. Most of this work was done really thanks to Faranek and her group. We did most of the analytical parts that I showed you, building statistical models, the in silico drug discovery platform, and also, I think the slides are going on their own. And also, the long organoid models were also built by one of my postdocs, joined with Faranek, Albertus Navickas. And with that, I'm happy to take your questions.
So I hope that gave you some time to submit some questions for the Q&A. So Hani has used the QuantSeq 3' mRNA-Seq to characterize the lung organelles that they have used to study the SARS-CoV infections. And the particular advantage of using QuantSeq for such kind of method is that it gives you a very quick assessment of the differential gene expression profiles. In contrast to mRNA sequencing, with QuantSeq, you only generate one fragment per transcript, and in the end, in data analysis, you only have to count the reads for each transcript. So that means QuantSeq is a very cost-efficient method to study expression profiling, in particular because it enables a high level of multiplexing. And the data analysis is straightforward and also included in the kits that we offer. On the second side, QuantSeq is also a very fast and robust method. Compared to conventional mRNA-Seq, you only need 5 major steps in this protocol of library preparation, and you don't need any pre-processing of the RNA, so no mRNA enrichment or ribosomal RNA depletion. So that saves you also a lot of time and also consumables. And if you want to go ever a step ahead and have hundreds or thousands of samples that you want to screen for expression profiles in parallel, we also offer QuantSeq-Pool. This is a very clever method where you can label up to 96 samples in the first step of the library preparation and pool them into one reaction, and then process all these samples in one tube for the downstream processes of the RNA of the library prep. So, and with that method, you can, in the end, increase the multiplexing to over 36,000 samples in parallel, if ever the capacities of NovaSeq are increasing that much. So that was just a brief overview over QuantSeq. If you want to know more about QuantSeq or QuantSeq-Pool, please contact us at support@lexogen.com or request an introductory offer from sales@lexogen.com, or visit our QuantSeq intro page that you can see here. And with that, I hand over back to Chiara and to moderate the Q&A session. Thank you.
Thank you, Martina. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd also like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. And so now we will start the Q&A portion of the webinar. The first question is, for Hani, these drugs would likely affect other genes like CYP19. How do they play a role in your model?
Very good question. So, I mean, these pathways, like central pathways like AR signaling, if you perturb them, you actually get a lot of gene expression modulations, that as I showed you in the RNA-Seq analysis as well. So the ACE2 was one among many that were significantly perturbed, so there's no question there. However, from a more clinical perspective, these drugs are all very safe. So despite the fact that they cause these changes are not central to physiology or homeostasis of the cells, and they don't really cause toxicity. So the viability of the cell does not depend on faithful expression of these genes, and how that might change in the presence of these drugs. So, and also what we are thinking about in the context of COVID-19 is actually a very short period of treatment. So that's kind of the short and long answer.
Okay. And did you test testosterone supplementation products that are used in men?
So I briefly showed DHT which is dihydrotestosterone. In basically a percentage of testosterone is converted to DHT. I think it's about 10% in the body, and it's just known to be more potent. That's what we use for some of our experiments that I showed.
Okay. And would anti-androgens also be helpful for treating women?
Yes, because well, first of all, women also have some testosterone, but also female hormones also feed into AR signaling. So AR signaling there are many paths into it, not just simply testosterone. Yeah.
Okay. And are anti-androgenic drugs something that are or would be used in the hospital for men with hypertension, diabetes, or obesity, or other conditions?
Yeah, exactly. So, anti-androgenic drugs are involved generally in prostatic diseases, and that's actually was some of the basis of our analyses for clinical outcomes, because we were basically looking at the kind of group of patients who were COVID-19 positive and also were likely to have received these drugs, basically.
And what would be the effective window of treatment in patients?
So this is basically the idea is that you're going to impact H2 expression, which is the receptor, right? So you want to, if you need to do this, you want to do it early, right? So bring down H2 levels on the cell surface so that infection slows down and doesn't go through and allows the immune system to catch up and clear the virus. But at the later stages, really COVID-19 is about basically less about the virus itself and more about the immune system. So at that point, it will be just simply too late. So there's the effective window would be kind of like early on in the process.
And what side effects do you think there might be given the importance of AC2 in regulating blood pressure?
So as I mentioned, these drugs have very good safety profiles, and they're actually taken for chronic diseases. And what we are talking about here, is the context of SARS-CoV-2 infection and COVID-19 infection is actually a very short period of time. So I don't think in the standard doses, which we have looked at, that would cause a problem.
Okay. Well, those are all the questions that we had come through. So we'll end that here. As a reminder, please look out for a survey after you log out of the webinar to provide your feedback. And if you missed any part of the webinar or would like to listen to it again, an archived version will be emailed to all attendees. And thank you for joining us for this Genome webinar.
Thank you.